Good day, everyone.
Good evening, Ma.
Good
Good evening.
evening. So it's nice to have our class again today. Last class, what happened? Nobody reminded me. Because I was so busy. I couldn't. But good evening.
Good evening.
Our last class, so it's nice to first reps have our class again today. Last class. Course reps, you did not remember to send reminder. This evening, I'll not take much of our time. We'll go straight to our topic for today. We've been talking about water. The last time we did um total water body weight. And we also talked on, who can remind me on the class we had the class before last week? Anybody has bothered to go to our materials, the right lecture materials? Anybody can remember? I also sent the lecture material for last class. So how we'll start tonight is I would like to list your questions. And for those of you who went through the material, how many of us went through the material? Please raise up your hand. Because I tried to make it as simple as possible. I tried to simplify it as much as I could. But if you have questions, please let us know. Otherwise, let's just dive straight into today's topic. So today we'll be talking about disorders of water and electrolyte balance. If we remember in our first class, we said total body water is 60% of the body weight, and it is less in female, in elderly, and more in infants. And I remember we calculated, each of us calculated total body water. Also, it's also important to note that the water must be balanced in the body. What do I mean? The water we take in must equal to the water that is excreted out of the body. And we take in water either through drinking, that's about 1.2 liters, through food, or through metabolism. Metabolism is the breakdown and synthesis of food we eat. Metabolic processes take place all of the time. As long as the person is alive, the body is in constant metabolic state. So we drink 1.2 liters. In food, we take in about 9 liters. And through metabolism, the body gets about 2 liters of water. Now, how do we let out water from the body? Either through urine, that's 1.5 liters, and is always regulated. ADH here stands for antidiuretic hormone. So ADH tightly regulates the water that leaves the body so that it guards against dehydration. And then insensible losses, either through sweat, vomiting, or what have you. 0.8 liters is lost through insensible. And then through feces, we go to the toilet every day. It's a normal thing for a healthy bowel. to be moved every day. So through feces, we lose 4.2 liters of water. Now, like I told us initially, ADH, which is also called antidiuretic hormone, is a hormone found on the top of the kidneys, like the cap. If you look closely, you see that like the kidney is wearing a cap. It's just a tiny cap on the kidney. So this hormone, when there is high blood osmolarity, it signals the hypothalamus in the brain. And what this does is that when ADH, when this high blood osmolarity signals the hypothalamus, it releases ADH, and then what ADH does is it makes the kidney's collecting duct to insert aquaporin 2, which is used to reabsorb water. You remember the function of the kidney, the loop of Henle and all the other parts. They actually help the body to retain water. They will release less urine when there is lower intake of water, when the water level in the body, in the blood is low. Now, what are the disorders? Some disorders of what of water in our body. We have the sodium disorders. And the first we'll be talking about is the hyponatremia. Hyponatremia. That is when the level of sodium is below 135. And this is how much, this also measures how much fluid is in the body. So hypovolemic, hyponatremia comes in three basic, could be that be hypovolemic, that is low volume dehydration, which is caused either through diuretics. Diuretics are things that we take that make us urinate. Things you take that will make you um use the toilet, either through purging or through constant urination. So we have hypovolemia when the water volume is low, which could be that through either vomiting. Euvolemic, that's normal water level, and hypervolemic when the water level is high. You either see edema, or if not controlled, it could lead to heart failure. So the key summary of sodium is that sodium flows along with water and it measures the amount of water in the body. So the urine is concentrated even in the if the bloodstream is dilute. That is, if there's no osmolarity, the urine becomes concentrated because water will not be let out. Now the next I'll be talking about is potassium disorders. Potassium disorder is the clinical name is hypokalemia. And this is a situation where potassium level is low, lesser than 3.5. The causes may be through vomiting, diuresis, or diabetes, lane shifts in diabetes. And this is the treatment, but I'll not delve on that. I just need us to focus on on the defects and what causes it. Now, this is hyperkalemia. Hyperkalemia from the word is high potassium. That is when the potassium level is greater than 5.0. And this might be as a result of renal failure, cell lysis, either through tumor or hemolysis, drugs, or acidosis. So that's what can cause hyperkalemia. Then we have calcium disorders. Please fill the poll, the attendance poll that is on now. I'll take it down any moment from now. So the next imbalance or water disorder is the calcium disorders. That's hypocalcemia, low calcium. When the level body calcium is less than 8.5. So these are the causes listed. We have vitamin D deficiency, pancreatitis, or hypoparathyroidism. Then hypercalcemia, when the calcium level is too high, maybe as a result of kidney stones or bone pains. So this table is giving us a quick check on the disorder type, and then for magnesium disorder, you have hypomagnesium. This is associated with hypokalemia and hypercalcemia. And then hypermagnesium is due to renal failure or magnesium contained in antacids. Antacids are what are used for some people have peptic ulcers. So they give them antacids. That's why you don't just take drugs at will. You take it by prescription. You don't just treat yourself because the body system must balance. That's the internal system of the body must be balanced. Any imbalance could lead to detrimental effects. So the next one we'll be talking about is the clinical pearls. That is key points to note. When there is weakness or low sodium or potassium in the body, the first sign might magnesium is absent or deficient. If an elderly person is confused or the level is high, it might be as a result of diabetes insipidus. So the list is there for us to take cognizance of. While we now go to how these electrolytes are calculated. You know, we were if one level is high or the other is low. Now, this is the formula for calculation of each of these levels. For similarity, the formula is shown above. Like I said, it's very important for all of these levels to follow because of keeping the body in good state. So it's this for formula for serum osmolarity. This is the anion gap. We were talking about that one in the next topic. So what our advice us is that we should know these formulas. They are not they are simple formulas. We should know them and practice worked examples. I try as much as I can to include examples and exercises in the lecture materials. But it's important to note that it is the sodium levels must be corrected if the sodium levels is low or too high. It must be corrected slowly to avoid cerebral edema because anything that happens to the brain most of the time is irreversible. So from what we have said so far, in a nutshell, is that water flows with sodium ion. So whenever there is an imbalance in sodium, the way it is treated by balancing the intake of water with the outflow. You'll notice that when somebody has been having diarrhea and vomiting, the first thing they will do is what? Who can tell us? When somebody has been having diarrhea and vomiting, what?
You,
Huh?
or oral rehydration saline.
Yes, oral rehydration, and immediately the person is admitted, they give him a drip, they put a line so that the body doesn't go into shock. The ORS, the salts in the ORS, ORS is just salt and water solution. NaCl, that's the formula for salt, is to replenish the sodium that has been lost, because sodium leaches with water. That's what this first point is saying. Water flows with sodium. Sodium flows with water, is water soluble. So what happens is immediately you give ORS, and when the person is admitted, the person is put on drip to put back the water that's been lost. Now, hypercalcemia can be fatal if not quickly handled. And then magnesium works in tandem with potassium and calcium. So when magnesium is low, it also affects the levels of calcium and potassium, and it's very hard to correct. So they say, the Englishman says, it's better to be safe than sorry. It's better to be safe than sorry. And then, because we say sodium leaches with water, even when if there be an imbalance in sodium, you don't just down the person, you don't just drown the person's system. You do it gradually, not too fast, so that the person doesn't go into the risks of osmotic demyelination syndrome, or the edema of the brain. So this is where I will stop for disorders of water imbalance. And the next topic I'll be taking us will be for today, because the holidays have been affecting our lecture time. And so we just have to move fast so that the other lecturer can come and teach his part. So the next we'll be talking about today: acidity, alkalinity, pH, and pKa, looking at the effects on cellular activities. Now, who can tell me what acidity is? Let's see how smart we are. Anybody? Acidity. Somebody should please give us a try. What is acidity? Or what is an acid? Nobody. Okay, to save our time, an acid is a proton donor. That is, an acid donates an electron, a proton, to to its neighboring, or in a solution. While the base is the proton acceptor. Now, weak acids weak acids are most important in human biology. An example of weak acids are as listed in the table. You have lactic acid and carbonic acid. While buffers, or weak bases, or their conjugates, is what is used in the biological system. Now, pH is the negative logarithm of hydrogen ion. And it is very important to note that the pH of the body of the blood is tightly regulated. It must not be too high or low. It must just fall within its range, because small changes in pH brings big effects. Then the next, the acidity, is the pKa, negative logarithm of dissociation constant. K means dissociation constant. That is how easily water or a chemical substance breaks down. That's dissociation. Okay, so pH at which an acid is 50% dissociated is pKa, and the best buffering. Buffering is resisting pH change. That's what buffers do. They resist pH change. The best buffering is when the pH equals to pKa. Okay, so I'll continue. Okay, so just what I said. Buffers, they resist pH change and keep things stable. Now, physiological values to reckon with. The gastric lumen, gastric means stomach, is usually between 1.5 to 3.5. This acidic, is this pH acidic or basic? 1.5 to 3.5. I didn't get that.
Acidic.
Thank you. So that's why when you don't eat, sometimes your stomach will be biting you, because the acid there in the stomach will start dissolving the stomach lining. You're having some pinching pains. So the gastric lumen is acidic, and it is so because it activates pepsin for digestion and also kills bacteria that we might have ingested in our food. Now, lysozymes is a weak acid between 4.5 to 5. And these are enzymes. Lysozymes are enzymes that break down cellular waste. Then the cytoplasm, the pH of the cytoplasm is 7.5. That's tending towards neutral. The pH of the blood is always between 7.3 and 4.5. And it must be maintained at this range, because any shift in pH, we said small shifts in pH have large effects. If there's a shift in pH of the blood, it can be very detrimental. Then the mitochondrial pH is 7.8, and this to maintain the proton gradient needed for ATP production. You know, ATP is the energy currency of the body. So all of these are tightly regulated, and a little shift in pH is not good for the body. So normal pH is always tightly controlled. Normal blood pH, if it is below 7.3, is the term called acidosis. If it's above, that's alkalosis. And the body uses 3 main systems to regulate pH. The body uses please mute your microphone. The body uses buffers to regulate pH. It uses respiration, as inhalation and exhale, our breathing, and it also uses the kidneys, the renal organs, to regulate pH. The Anderson equation is often used to calculate the level of acid to base in the body. Please, these equations will come out in our either in course or exams. Advise us to learn them. So this Henderson-Hausenbach equation, we said, is used to know the ratio of acid bicarbonate buffers to base in the body, in the blood. And an example is given below. These are the values given. Blood pH is 7.4 and the pKa is 6.1. Now the equation states that pH is equals to what? pKa plus log of bicarbonate. HCO3 is bicarbonate over what's H2CO3? Base, acid over base. So let's see. The value given for blood for pH is what? 7.4 is inserted. And the value given for pKa of carbonic acid, that's H2CO3, of carbonic acid is what? 6.1. So when we insert the values and collect like things, the log of base over acid is equals to what? 1.3. And then converting 1.3 into logarithm, it gives us 20. So for the ratio of base to acid, that's 20 to 1. That's the ratio of bicarbonate to carbonic acid. And please take note of this ratio. This ratio is very crucial in human physiology because this ratio keeps the blood stable. The ratio keeps the blood pH stable. Excuse me, please, just a minute. Also, small changes, like we said initially, can cause significant shifts in pH. So the ratio is key. And the body works really hard through the lungs, kidneys, and buffers to maintain a stable ratio. Now, the buffer systems. We said the buffer systems help the body to resist pH change. The buffer system the body uses are bicarbonates, phosphates, and proteins. So you can see why it's important to eat proteins in your meals. The pKa for bicarbonate is 6.1, and it is found in extracellular fluid. It is most useful buffer in the body because it's an open system. The lungs remove CO2, kidney adjusts HCO3. HCO3 is bicarbonate. Then we have phosphates, is the next buffer in the body. Phosphate is found in intracellular fluids and urine. And then its role is important in urine to excrete acids because the acid level of the body must not be too high, must not be too high, otherwise you have acidosis or alkalosis. Now, protein, especially histidine. Histidine, is it an essential amino acid or a non-essential? It's an essential amino acid, so we must get it from our proteins. Histidine is found in the red blood cells and plasma proteins. And hemoglobin is the major buffer in the red blood cells. So these three buffers is what the body uses to resist changing pH. We told, if you remember, I said the blood pH must be, what's the range for the blood pH? You can remind us. 7 point what?
7.5 to
7.5.
7.45.
35, thank you. Between 7.35 and 7.45. It must not be lower or higher than that. So these buffers are what the body uses to maintain its pH. Now, effect on cellular activities like enzymes. pH changes alters the charge. pH, the charge on amino acids in enzymes. And then when this happens, it affects substrate binding. If we remember factors that affect enzyme catalysis, one is pH, one is temperature. There are about 5 of them. So what happens is that when there is a pH change, we said small change causes big effect. Always remember that small change in pH causes big effect.
Hello?
And this is
What
the
did you
reason.
say?
The reason is because once there is a change in pH, the enzymes cannot work as they should. Extreme pH change can in nature permanently damage proteins. And all enzymes are proteins. So these are enzymes. Pepsin is in the stomach. Salivary amylase is in the mouth. That's why carbohydrate digestion starts from the mouth. You have trypsin, and the list is there, and they are pH, at which they work best. If any of these pH is altered, these enzymes are denatured and cannot function. The next is the protein charge and isoelectric point. Isoelectric point, which is termed Pi, is the pH at which protein has a net charge of 0. Below Pi, protein is positively charged. Above Pi, protein is negatively charged. So for albumin, if the Pi of albumin is 4.7, what this means is that it is strongly negative at normal pH, and when it happens, it binds calcium, drugs, and fatty acids. And whatever chelates calcium, whatever binds calcium, makes it to be unavailable in the body, and so will cause some damages. Then we go to membrane transport. That is ion trapping. The membranes are semi-permeable. They are highly selective of what goes into the cells or what goes out of the cell. So, talking about ion trapping and pH partitioning hypothesis. For weak acids like aspirin, what is aspirin used for? Who can remind us? For weak acid like aspirin, the pKa is 3.5. And since the pH of the stomach is 2, it is easily absorbed within the stomach walls. So it helps it to do its work. So, but in the blood, weak acids are deprotonated because the blood pH is 7.4. So weak acids are deprotonated, and then they are low and they become trapped. Do we understand the message I'm trying? In toxicology, we talk about transport for drugs. That's why you see that some drugs are put in capsules so that they can get to the point where they are needed. This membrane transport is just trying to make us understand that depending on the medium of the physiological part of the body. Like in the stomach now, the pH is acidic. If the drug is aspirin, it will be highly easily absorbed because they are within the same range. But when we're talking about weak acids and in the blood, they are miles apart. One is an acid, one is one is tending towards a base. So the pH is, the acid will be trapped because it will be deprotonated. We said acids are proton donors. That was how we started the class. Acid donates protons. And then we have the list goes. So the clinical application is that if there is an overdose, take for example, aspirin, it will be alkalized in the urine and excreted faster. Now let's go to calcium and pH. Acidosis and alkalosis. Acidosis happens when the blood pH is low and there is increase in calcium. Alkalosis is when the blood pH is high and there's a decrease in calcium. It's important to note that pH affects almost everything in the body: enzyme, drug absorption, oxygen, electrolyte balance. So maintaining the blood pH is crucial for life. Now, how do we maintain blood pH? Who can help us? How can one maintain his blood pH? Any idea?
And making sure that the electrolyte leaving the body is equal to the electrolyte.
Thank you. By making sure that input equals output. When you see that you are having diarrhea or vomiting, you don't wait until you collapse. You give yourself first aid. And if you don't wait until things get out of hand before you now start looking for what to do. Salt water solution. If there's no, if somebody is purging and there's no salt water solution, what is an alternative that somebody can use? Who needs? What is an alternative? And that Gary water. You know Gary now. This is Nigeria. Do we know Gary? Class. Who does not know?
You know, Gary.
So once you just soak Gary, if there's
We
no
know Gary, man, especially Jebu Gary.
I didn't say Jebu Gary. All you need to do is soak. If the person has been purging and has lost too much fluid, and there's no ORS around, just soak Gary in water and drink. Not, I didn't say drink Gary. Drink the water of the Gary to help to put some electrolyte before the person can be quickly rushed to the hospital attended to. But if the person is vomiting, the earlier you get to the hospital, the better. There is a shift in pH or before it's dehydrated. So main acid-base disorders. The main acid-base disorders we have are metabolic acidosis. And metabolic acidosis happens when the blood pH is lower than 7.3. Primary effects is that there's low bicarbonate. And then what happens, how the body tries to compensate is through the breathing. It's called the Casmal breathing, hyperventilation. The person will be breathing, having like fast, breathing very fast. You know, there's a normal way to breathe. But when it's hyperventilating, it will have if it's not quickly handled, it will can affect any physiological organ in the body. Then the next acid-base disorder we have is metabolic alkalosis. And this happens when the pH of the blood is higher. There's an increase in bicarbonate, and how the body tries to wriggle around this is through hypoventilation. We have respiratory acidosis, same pH value, but this time respiration, the kidney is the one that handles it, and the kidney retains water, doesn't allow water to leave the body. And you have respiratory alkalosis, low bicarbonate, and the kidney excretes more water. So for metabolic disorders, the lung compensates. For respiratory, the kidney compensates. Now, the next we go to is the anion gap. Anion gap. We've talked so much about acid bases, weak bases. Okay. Now, what is an anion? Anion gap helps one to detect unmeasured anions. That's the unmeasured acids in the blood. And it's very important because it helps to evaluate the levels of metabolic acidosis. So Ag, which is the anion gap, the formula for calculating this is sodium ion minus chlorine ion plus bicarbonate ion, plus bicarbonate gives us the levels of anion in or free or measured acids in the blood. This is the normal range, and it just helps to buttress the point we've been saying before. The blood pH must not shift. The body pH must not shift. Because if the gap is high, it means there is more acid present in the system. And which is why, all these drinks, all these sugar drinks, sugar dehydrates, sugar drains fluid out of you. So it's always better, even when you're thirsty, it's better to drink water than to load your system with sugar. A lot of us, we think we're enjoying, but the truth is all these processed things we take and highly sugary and carbonated things we take, they do more harm to our system than good. So it's good for us to take note. Drink more water, keep your body healthy and out of trouble. Now, high anion gap and metabolic acidosis seems to be the most clinical situation, which is why the MOD files is an acronym, is used to denote the high anion gap. For M means it's caused by methanol, toxic alcohol, or formic acid. U is caused by uremia, that's from severe kidney failure. D, diabetic ketoacidosis, is as a result of ketone bodies. When the body cannot, when carbohydrate is absent, fat is converted to ketone bodies. But when the ketone bodies, the blood is too high, that's when you have DKA, diabetic ketoacidosis. So This is the meaning of the acronym DUPILE. And each of these letters is showing us what causes, what's the clinical cause and the physiological state. How is this, how to use anion clinically? Okay. This is just for our information because this is not a clinical class. I would just like us to focus majorly on the electrolytes and the organs affected or what is causing it. So what I would like us to take from here is that high anion gaps signifies the presence of unmeasured acids. And most common causes in real life are lactic acidosis, decay, and uremia, which we've seen in the table. Now, DKA. DKA is important because of the sugar levels in most of our processed beverages and foods. There's a lot of diabetic situation, both young and old. So DKA, keto acid production from fats, breaks down in insulin deficiency. What happens in diabetes is that the body cannot metabolize. The body has a challenge with carbohydrates, sugars. So even though the person eats, it cannot handle carbohydrates. And so what happens is it begins to break down its fat stores, converting them to ketone bodies, whereas there's carbohydrate in the blood already. So what happens is now that once ketone, the levels of ketone rise, it results in diabetic ketoacidosis. I just noticed that I've been the only one talking since the beginning of this class. Is there any questions so far what we do not get in the course of the lecture? Does anyone have a question? Either you unmute and ask, or you raise up your electronic hand. Can we continue?
Yes, ma'am.
Yes, ma'am.
So you understand everything. Me, I will ask my question at the end of the day, trust me. So the next clinical state is the hyperventilation tetany. And in this, what happens is hyperventilation leads to reduced PCO2, reduce the levels of sodium, CO2. What's CO2? Carbon dioxide. Reduce level of carbon dioxide, which results in respiratory alkalosis. And when this happens, more calcium binds to albumin, and there is neuromuscular irritability. Small change in pH leads to what? Big problem. Then there's this aspirin overdose. Aspirin aspirin is like a painkiller that is abused, is often abused. So what happens in aspirin overdose is that because aspirin, we saw the pH of aspirin is acidic, it contributes to metabolic acidosis. and leads because it increases the pH in the system, in the stomach. Lysosomal dysfunction. Just as we saw in the table, all this is an explanation of what I've explained initially in the table, and this is appearing here again. So, from our class of today, the essential formulas have helped us to keep them in a table. We had the pH is what's negative logarithm of hydrogen ion concentration, and that's converting hydrogen ion to pH. We have pH equals pKa plus logarithm of acid over base. That's the Anderson-Heisenberg's equation for calculating buffer. And we have the Ag, anion, what's Ag again? Anion gap. Helps us to detect unmeasured anions. Please know all these formulas because you will meet questions relating to these formulas. And don't just know them, also work questions on them. Also practice questions. So far, what we've discussed on acid, bases, and buffers is that pH is logarithmic. That is, small changes in pH means massive change in hydrogen ion concentration. From that example we did, you could see when we took 1.3 to the logarithmic form, it gave us 20. So pH is logarithmic, meaning small changes in pH leads to massive change in hydrogen ion. When pH equals pKa, buffer is the strongest, that is 50% dissociated. Weak acid and bases dominate biology and physiological system because that is where buffers work effectively. I will say buffers. What are buffers? Who can remind me? What are buffers? Buffers
Any substance
that resist change in pH. Thank you, everyone. Buffers are substances that are used to resist change in pH, and I told us the three buffers that the body uses and the three types of buffers. Okay, so pH controls enzyme activities, protein charge, drug absorption, oxygen delivery, and ATP synthesis. So pH controls, in short, every metabolic activity in the body. That's why small change in pH results in massive problem. And the blood pH is always between 7.35 and 7.45. It's not negotiable. It must not shift. Otherwise, it could lead to cellular dysfunction and death if not quickly handled. Any question for the lecturer? So from next week, Dr. Lawal will be the one taking the, and then when Dr. Lawal is done, when it's time for in-course, you'll combine both and you have your in-course. In the absence of question, please let us share the grace. May the grace, let's all unmute and share the grace. May the grace of our Lord
of our
Jesus
Lord
Christ,
Jesus
Christ,
Christ,
Jesus
the
the
love
love
Christ,
of
of
God,
God,
love of God,
and the fellowship of the
and
Holy
the fellowship of the Holy
Spirit
Spirit,
is
Spirit,
now
rest and
and
abide
remain
with us
now
now
forevermore.
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 Amen.
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 thank you for your time.
God
Thank you,
Thank
Ma.
you,
bless
ma.
everyone.
Bye, Ma.
Thank you, Ma.
Thank you.